All right, so chapter 14 is going to focus on Mendelian genetics. So, what genetic principles account for the passing of traits from parents to offspring? The blending hypothesis was disproved by Mendel. The idea that two parents provide genetic material and that those particular pieces of genetic material basically blend. And then there was also the particulate hypothesis that parents are able to pass on discrete heritable units known as genes. And that is the one that Mendel was able to provide evidence to support. As we talked about in class, we know that this particulate hypothesis is a fairly simplistic way to look at genetics, that it definitely is more complex, but the data that has been gathered does not disprove Mendel's hypothesis and the idea that parents can pass on genes to their offspring. And this allows traits to disappear and then reappear in subsequent generations. And he looked at this with peas. So through his peas, he was able to identify two laws of inheritance. He bred garden peas because there were very distinct characters and variants of those characters known as traits that could be observed. So characters is just the general type of feature being observed, and then the traits would be the specific observations of those characters. You could control mating with peas because each of the flowers had both a stamen, which would be the sperm-producing organs, and an egg-producing organ known as the carpel. So you could remove the stamens from one of your pea plants and use stamens from another pea plant to fertilize those carpels. So cross-pollination is pretty straightforward. And so there's just showing you that happening. He wanted to only look at characters that were able to be observed using two distinct forms. So two different types of flower color, two types of seed color, things like that. He was able to do this by creating varieties that were true breeding, that were homozygous for the trait that they were showing. They only produced offspring of their variety if they were self-pollinated. And when two contrasting true breeding varieties were mated, hybridization was what took place. When the true breeding varieties are known as the P generation, and the plants that were produced from that hybridization process were known as the F1 generation. Any F1 plants that are then either self or cross-pollinated would result in F2 generation. If they were cross-pollinated, they would need to be cross-pollinated with another F1 plant. So here is an example where he did this with flowers. He had purple flower parents and white flower parents, and what he saw in the F1 hybrids was that they only produce purple flower plants, but the F2 generation, which was produced from your F1 generation, produced primarily purple flower plants, but also white flower plants in a ratio of about 3-to-1. So Mendel's thought was that the purple flower was the primary factor affecting the flower color in your F1 hybrids, and that color was known as a dominant trait, while the white flower color that was not seen in the F1 generation was a recessive trait. The alleles, the factors for the white factors, was still there. It just didn't show up in F1 because you didn't have both factors from mom and dad. And so he looked at this with 6 other plant characters, and we now know that those heritable factors are known as genes. So there are the ones that he looked at: flower color, flower position, seed color, seed shape, pod color, and stem length. As you can tell from the ratios, none of them are exactly 3-to-1 because this is based on experimental data, and the 3-to-1 ratio is the ideal ratio if everything went perfectly. So Mendel's hypothesis to describe that 3-to-1 ratio was based on 4 concepts that are used to describe it. And the first one is that we have alternate versions of genes, which are what are going to allow us to have variations in our characters, and that these alternate versions of genes are known as alleles, and they are located at specific locus on chromosomes. So your homologs each are going to contain a locus for flower color gene with these Ps. The second is that for each of these characters, organisms obtain 2 alleles, one from mom and one from dad. Now keep in mind, Mendel didn't know about chromosomes at the time, so all he was describing was that there was genetic information being passed from each parent to their offspring. If the alleles were identical, then you would have true breeding plants. If they were different, you had the F1 hybrids. The third was that if you have the different alleles at a locus, a dominant and a recessive allele, the dominant allele is going to determine the phenotypic expression, what we know of as now, the organism's appearance, while the recessive allele is not. So that's why with the F1 generation, we only saw purple flowered plants. The fourth, the law of segregation, is that the alleles for these characters are going to separate during meiosis and end up in different gametes. So you get the parents have 2 alleles on their homologous chromosomes. One copy of one of the homologous chromosomes is going to end up in one gamete, an egg or a sperm. The other one would end up in another gamete. And so that corresponds with what we've talked about previously with meiosis and how the homologous chromosomes are separated. We can use Punnett squares to explain these relationships. You probably have done a lot of Punnett squares previously in your first biology class, where you show the different combinations of alleles that can be passed on by the sperm and the egg, by the male and female parents in that particular cross, and you can predict what the offspring would be. When we use capital letters, we're dealing with dominant alleles. When we use lowercase letters, we're dealing with recessive alleles. And so there you can see the Punnett square for crossing the 2 F1 plants. And you can see what the offspring would have been for F1 based on that parent generation, in that the purple flowers could only give a dominant P allele, while the white flowers could give a recessive P allele. So some vocabulary that hopefully is review. Homozygous is when both alleles are identical. Heterozygous is when you have different alleles for a gene. Heterozygotes cannot be true breeding. Homozygotes are. You can have homozygotes that are dominant and homozygotes that are recessive. These alleles are not always going to be able to be ascertained by their physical appearance. So phenotypes can differ from genotypes. Phenotypes would be your physical appearance. Genotypes would be the allele combination that you have on your chromosomes. Why we say that phenotype is not necessarily going to match up with genotype. If you have homozygous dominant as your alleles or heterozygous, you would see the same phenotypic expression according to Mendel's laws. And there's just showing you that genotype versus phenotype. So how could we determine what was the genotype of an individual that is showing the dominant trait? We would have to perform a test cross using an individual that contains the recessive alleles for that particular trait. If all of the offspring show the dominant trait, have the dominant phenotype, that tells us that the parent must have been homozygous dominant for that allele combination. If any of the offspring show the recessive phenotype, that tells us that the unknown parent was heterozygous for that particular trait. So there you can kind of see the examples with test crosses. So Mendel figured out or described the law of segregation using a single character. Those would be known as monohybrid crosses. His second law of inheritance was determined looking at two characters simultaneously. So he crossed two true breeding parents that differed for two different characters and produced F1 offspring that were heterozygous for both of those characters. And then when a dihybrid cross was performed between those F1 dihybrids, we were able to ascertain whether the characters were passing independently or whether if the F1 parent had homozygous alleles for one and homozygous alleles for another if they stayed intact as they went into their offspring. And so there you can see the original parents who could only give the dominant alleles for the yellow rounded seed and then the recessive alleles for the green wrinkled seed. The F1 generation was recessive for both of those traits. And if those two had truly stayed together as a unit, then you would have only had Y with R and little y with little r. And you would have only had a 3-to-1 combination. You would have only seen the traits that you observed in the parents back in the P generation. What we now know to be the case is that the independent assortment hypothesis was supported. The data that was collected with those pea plants showed that you can have different combinations of alleles and different phenotypic expressions that don't necessarily match up with any of the phenotypic expressions or the allele combinations that were present in either the F1 generation or in the P generation. And so that ratio is 9:3 to 3:1, 9 showing both dominant alleles, or showing both dominant phenotypes, and then 3/16 of the time showing one of the dominant traits but recessive for the other, and then 1/16 of the time showing both recessive alleles. And so this independent assortment is that every pair of alleles is able to segregate independently of another pair of alleles when these gametes are formed in meiosis. This again is only going to apply to genes that are not linked. And if they are not linked, they are either far apart on the same chromosome or they are on completely different chromosomes. If you happen to have a series of alleles that are close to one another, and we'll spend more time on this in chapter 15, they tend to come together as a unit. But if they are far enough apart that they can be segregated independently in meiosis, then they are. So probability is going to play a big role in how we do Mendelian problems because we won't necessarily just be looking at one trait or two trait. We won't necessarily want to know about every possible characteristic for the combination of individuals that we are using to produce offspring. So probability allows us to examine only the options that we want to truly look at. We know through segregation that the homologous chromosomes are segregated independently from one another, and that alleles that are being passed on through those chromosomes are the alleles that are found on mom's chromosome 1 don't necessarily follow the alleles that were found on mom's chromosome 2. You can have different combinations of chromosomes and you are not necessarily going to get the same combination of, you're not going to get both, you're not going to get mom's chromosome 1 and dad's chromosome 1 in the same gamete. You're only going to get one of those two. So the multiplication rule is that if you are looking at two or more independent events, and you want to know if what the probability was of both of them taking place at the same time, you would multiply their individual probabilities together. So we would be able to do this in an F1 cross, Each gamete has, if you have a heterozygous plant, has a 1 in 2 chance of getting the dominant allele and a 1 in 2 chance of getting the recessive allele. And so if we combine those probabilities together, 1/2 times 1/2, the possibility of getting both dominant alleles would be a 1 in 4 chance. The possibility of getting both recessive alleles would be a 1 in 4 chance. And you have 2/4 of a chance of getting a dominant and a recessive allele, because you could get one from mom, the dominant allele, and the recessive allele from dad, and vice versa. And that would be the addition rule, is that if you have one or two more exclusive events that can take place, like we just saw there with the recessive alleles, you can add their individual probabilities together. So this is going to make life, again, a little bit easier when we're getting into more complex genetic problems. So as I said, we're going to look at this with dihybrid. You want to look at the different combinations for your characters, and then you can multiply them together when you're looking at multiple traits at a time. So what is the probability of getting big Y? With this particular, given those two gametes that have big Y, big R, little y, you have a 1 in 2 chance of getting big Y from your mom and a 1 in 2 chance of getting big Y from your dad. So you have a 1/4 chance of getting both dominant alleles. And the same with the R's. So multiplying those two probabilities together, we give you a 1 in 16th chance. Because you can get big Y from both parents and little y from both parents, the same with big R and little r, you have a 1 in 2 chance of getting big Y, little y from your parents, but you can only get big R one of the 4 times, since both parents have to have the big R and both of your parents only have one of them. So you have a 1 in 2 chance of getting the big R together. That would give you 1/4. And so the problem below, you have two other gametes with 3 traits, heterozygous for P, one heterozygous for Y, and homozygous recessive for the other. The other one's homozygous recessive for Y. And then finally, the gamete that had 2 heterozygous sets of traits is homozygous recessive for little r, and the other gamete would be heterozygous for R. So looking at the combinations that are given there, you want to know what possibility it is that these gametes are going to produce offspring with 2 recessive traits. So you would want to determine what genotypes could give you 2 potential recessive traits at a minimum. You could have P and little p, little y, little p, little r, little y, little r. And then keep in mind, you could also have, because of the gametes that are present, you could have heterozygous for P, and you could have homozygous dominant for P. You could also have homozygous recessive for all 3 sets of alleles. Why is it that you can't have little p, little y, little big R? Because one of your gametes did not have any dominant R alleles. And it would be the same way with Y. So because P, both gametes were heterozygous for those sets of alleles, that's why you had the 3 different combinations for P with the recessive alleles Y and R. So you would go through your probabilities. What are the chances you could get those combination of alleles? Again, take a look at each gamete. So for the first gamete, we have a 1 in 2 chance of getting little p. For the second gamete, we have a 1 in 2 chance of getting little p. 1/2 times 1/2 would give me 1/4. For the Y alleles, we need both to be recessive. You have a 1 in 2 chance of getting the little y allele from the first gamete, but you have only can get the little y from the second gamete. 1/2 times 1 would give you a 1/2 probability of getting little y as those set of alleles. We would do the same thing with big R, little r. You have a 1 in 2 chance of getting big R from the second gamete. You can only get a little r from the first gamete, so there's not the second option available to you to look at. You only would have a 1 in 2 chance of getting that combination. Multiplying those probabilities together would give you a 1 in 16th chance of getting that particular combination of alleles. You would look through these other examples exactly like that, And once you have the individual probabilities of those combinations of alleles, you can determine the chance of having two recessive traits by adding all of those individual probabilities together. So the inheritance patterns that we've been talking about so far are oftentimes more complex than what Mendelian attempts to describe. It works great for pea plants, but we know now that there are heritable characters that are not going to be determined just using a single gene and not necessarily with two alleles. The idea of segregation and independent assortment is still, those ideas are still going to play critical roles, however, in these more complex genetics problems. So if we extend Mendelian genetics just looking at a single gene, we're just going to focus on that right now. Sometimes we have situations where we have alleles that aren't completely dominant or recessive. Sometimes we have genes that don't just generate two alleles, they could generate more than two. And sometimes we have genes that end up showing multiple phenotypes. So in terms of dominance, complete dominance is what Mendel's work showed, that the heterozygous form of that combination of alleles and the dominant homozygote were going to show the same exact phenotypic expression. When you have incomplete dominance, you have a blend, an intermediate between the dominant phenotype and the recessive phenotype. With codominance, you actually are going to show both sets of both traits that you would see with dominant and with recessive. So I know here we're showing just the incomplete dominance form with that particular picture, showing that the heterozygous form of snapdragons would produce pink snapdragons. If the color of snapdragons was codominant, you would expect to see both red and white on the flowers that are being shown in this picture. We see codominance, for example, with blood type, because you can see both A and B alleles. You can have A blood, you can have B blood, and you can have AB blood. So the dominant alleles don't just kind of shut down the recessive alleles. They're just different variations in the order of the nucleotides present in your DNA. And it really is just dependent on how we examine the phenotype as to what types of relationships we see between dominance and recessiveness. So some examples of real-life applications of this would be with Tay-Sachs disease. It is fatal. It causes lipids to build up in your brain. So at the organismal level, we have a recessive allele, but at the biochemical level, we have the enzyme activity. It's not that the enzyme isn't there, it's just not functioning properly. So that would be an example of incomplete dominance. Dominant alleles aren't necessarily going to be seen more often than recessive alleles, because sometimes you really don't want to have the dominant, you would hope that the dominant alleles would not be passed on. One baby out of 400 in the US is born with extra fingers or toes. This trait happens to be dominant, so we are quite happy that the recessive alleles are present in greater abundances than those dominant alleles. Said multiple alleles. Genes can exist in populations that have more than two allelic forms. Blood type, which is going to determine which carbohydrates are attached to your blood cells. There are 4 different phenotypes that can be seen in human blood. You can have A blood, B blood, O blood, and AB blood. The capital I with the superscript A and the capital I with superscript B are showing that those alleles are codominant, while the little I is showing the recessive allele. And when you have two little I alleles, you would have the O blood type. So the IA allele codes for the A carbohydrate, IB allele codes for the IB carbohydrate, and when you have both of those, you get both types of carbohydrates. If you have I, you don't get any carbohydrates that are going to be attached to your blood cells. And that's how your cells are able to recognize those different blood types. Pleiotropy is when you have genes that are going to have multiple impacts. And so, one example of this would be with cystic fibrosis, another would be with sickle cell disease, where you can have alleles that are going to have an impact on more than one physiological process. So, with sickle cell, you have a single gene that has a mutation, and pleiotropy is going to show how, if you have sickle cell, you could have an impact on your sight, on your liver, as well as on your heart. Again, Mendelian genetics can be extended beyond a single gene. It can be examined looking at two or more genes. And epistasis would be an application of that, where a gene at one locus is going to impact on the phenotypic expression of a gene at a second locus. So for some mammals, the color of the coat of that mammal is dependent on two genes. One gene determines the actual color, and the other one, the pigment color, while the other one determines whether the pigment will actually be deposited, will be added to the hair. So here you have two Labradors that are heterozygous for those two alleles, and you see the different combinations of alleles that could be passed on via meiosis to their offspring. And you see that for the majority of them, they're going to have a color to their coat. But if they got both recessive alleles for the deposition of that color into their coat, no matter what set of B alleles they got, they are not able to show that black or brown color in their coat. They would be your golden Labradors. Polygenic inheritance is going to have an example that would be skin color, where you have a variation of these characters along a range, a continuum. And so typically more than one gene is having an impact on a physical expression that we would see, a phenotype. So here we see that at least at this time we know of 6 different combinations of alleles, 6 different genes, that depending on what alleles you have from your mother and father are going to have an impact when you combine them all together in terms of your skin tone. So lots of different potential combinations. There are 6 different, it says, phenotypic expressions that we would see depending on these combinations of alleles. Crazy. So nature and nurture, how environment can impact on your phenotype. Sometimes these characters are going to be dependent on your environment. The norm of reaction would be basically your phenotypic range that's influenced on your environment. Hydrangea flowers can vary from pink to blue-violet. It depends on how acidic or how basic your soil is. The more genes that are involved with these characters, the broader these norms will be. And so they are considered to be multifactorial characters because not just the alleles affect your phenotype, but the environmental conditions do as well. And together in combination, they are going to have an impact on your phenotype. So organism's phenotype is going to involve the physical appearance, but it also can have an impact on your anatomy, on your behavior, on your physiology. And so phenotypes are not just based on your genotypes, they're also based on your environmental circumstances. A lot of human traits follow these Mendelian patterns, but humans are not going to be the greatest organism to use to study genetic research. The word I could not come up with in class. The ethics of studying humans would be a big consequence, but more practical reasons, that's on its own is enough of a reason not to. But humans have a very long gestation period. The time it takes to produce offspring is 40 weeks. Um, that takes quite a lot of time to be able to conduct research. Typically, only one, possibly two offspring are going to be produced. That's not going to help a lot when you're looking at these probability ratios. And again, the last one is by far, forgetting the first two, the last one pretty much takes care of it. But we can examine genetics in humans by looking at relationships between parents and offspring, and siblings of parents and grandparents of offspring. So we can examine relationships using um, the families that are already present. And so pedigrees are often used to be able to look at these relationships between parents and children across generations. And we can determine based on those pedigrees inheritance patterns, whether alleles are being passed through your non-sex chromosomes, they're autosomal, whether they are being passed through sex chromosomes, and they would be X-linked, since both um, females and males get an X chromosome, whether the pattern of inheritance is based on dominant alleles or whether it's based on recessive alleles. This can also help us better ascertain what potential combinations we might see in offspring. So here we have widow's peak, and you've got your key at the top. Males are the boxes, females are the circles. The filled in ones are the individuals that have the particular trait. When a line is connecting the boxes, that is a couple that has produced offspring, and then their offspring would come off of that particular line. So we're looking at widow's peak. We see that the third generation, we have two females, one that had widow's peak and one that did not. We see in the second generation that the parents of the offspring both had widow's peak, and that one of their parents had a widow's peak. But we see that of that first generation where we had one of the two parents with widow's peak, only half of their children showed that particular trait. So is widow's peak a dominant or recessive trait? I know that they're labeled with the capital alleles and the lowercase alleles. If we have a parent that is not carrying the trait, and we are seeing things split 50-50. We know that from test crosses, if we're only seeing that allele show up half of the time, that our original parent was heterozygous for that particular trait. What makes it kind of interesting in the second generation, since we had two parents marry, and they both were showing that widow's peak phenotype, they produced offspring. We've got each with a dominant allele, and we have in theory, three of the four should show that widow's peak trait. Because they still had that recessive allele, we have no widow's peak being shown with one of the two siblings. If they had continued to have siblings, we would have expected probably more of the siblings to show that widow's peak trait, because all you had to do was be heterozygous to show it. So widow's peak would be a dominant trait. If you have one of those dominant alleles, you're going to get that particular trait. Over here on the right-hand side, we have attached earlobes. We have, in the third generation, an individual that has both recessive alleles, and we have her in that, I don't know if it's a guy or a girl, it looks like a girl to me, but the other individual does not have an attached earlobe. They have a free earlobe. So if we work our way back, we see that neither parent had attached earlobes. That right there is kind of a giveaway, because when neither parent has that attached earlobe, but one of their offspring does, the trait disappeared and then showed back up again. And that's most likely because it was a recessive trait. If we keep working our way back, we see that in the first generation, we had on the right-hand side, we had a parent that had an attached earlobe and one that did not, and they produced two children, one that did and one that did not. While the other side, with the second generation, we had two grandparents that were recessive for those alleles, and it was approximately a 50-50 split between the dominant genotype and the recessive genotype, 2 out of 4. Probably would have expected it to be more 3-to-1, but again, this is why you can't say absolutes when it comes to genetics. The fact that the trait disappeared and then showed back up would be why that this particular pedigree is showing attached earlobe as a recessive trait, while over on the far left, the fact that we had the dominant alleles present, that the parents continue to produce dominant allele, the traits are not disappearing, that's what would be an indication that this is a dominant trait. A lot of genetic disorders, however, are inherited recessively. Sometimes these disorders are not going to have a big impact on that individual's life, and other times they will. You're only typically going to see these recessively inherited disorders when individuals are homozygous for those alleles. Carriers would be your heterozygous individuals. In general, they tend to be, you would not see any difference in their phenotype. And so if you have offspring that carry a recessive disorder, their parents most likely were heterozygous for those alleles. Albinism is a recessive condition, so you would have parents that both carry the recessive allele for this, that results in a lack of pigmentation. So there you've got two normal parents, you see the different combinations of alleles that could come from those parents, and then you see the siblings, one of which was just albino and the other one which is normal. Whether they are heterozygous or homozygous dominant, there would have to be more genetic testing done to determine that. You would have to do some sequencing with the DNA. If a recessive allele causes a disease is rare, it's pretty unlikely that you're going to end up with, through random mating, two individuals that are going to mate and produce an individual that carries both recessive alleles. This will happen more frequently if there is not as random of mating, if there is marriage between closer relatives, which is why in most societies, marrying between marriages between first cousins, having children between first cousins, having children with siblings, children with direct relatives is highly disapproved. Cystic fibrosis is the most common lethal genetic disease we have here in the US, one of about 2,500 individuals that have a European background are likely to end up with cystic fibrosis. This allele, when it's not functioning properly or if it's just not present, will cause chloride transport channels not to be there. Sorry, the cystic fibrosis allele is always there. I misread part of this. Too bad. When that allele is recessive, you can have transport channels that will not function properly in terms of moving chlorine, or they just won't be there. And so that's going to cause chloride ions to kind of build up outside of your cell. And so when you don't have those chloride ions being able to kind of move when they need to from in and out, some of the physical manifestations of that are mucus buildup and nutrient malabsorption. I already talked about sickle cell a little bit already. This affects approximately 1 out of 400 African Americans. It is due to a single amino acid that is substituted in the hemoglobin protein. And when you are homozygous for sickle cell, all of your hemoglobin is abnormal. So some physical manifestations of that include organ damage, pain, paralysis, physical weakness. If you are heterozygous, typically you're healthy, but you can have some symptoms. And what is pretty wild about this, 1 out of 10 African Americans tends to have the sickle cell trait, which is pretty high. But if you are heterozygous for this particular disease, you are going to be able to be less susceptible to malaria. So, although sickle cell is not something that any individual would want to have alleles for, that is one positive out of having an allele for sickle cell. Dominant inherited diseases, again, you have to have just at least one of those dominant alleles for that to happen. Dwarfism is one example of that. Okay. If your parents, if one of them was a dwarf and the other one was not, you've got a 50-50 chance of having an offspring that is also a dwarf, because all you need are one of those alleles. So it's kind of like Huntingtons when we went over it in class. It's highly unlikely to end up with both dominant alleles for a trait that is passed in a dominant manner. But all you need is one of them to show that trait. So Huntington's is a degenerative disease of the nervous system. You don't typically see any issues with Huntington's until you're a little older, 35, 40 years. Once the nervous system starts to deteriorate, you're more likely to be examined to determine if you have it. It is irreversible and fatal. So there you can see with the individuals, autosomal dominant, you have a parent who has this particular disease. And by the time you're 35, 40, a lot of times you've already had children by that point. Your children typically are going to have a 50-50 chance of getting Huntington's, because the parent who is afflicted with it was most likely going to either give a dominant or recessive allele. Again, most of the time with these autosomal dominant traits, you're only going to be heterozygous for that dominant trait. Other disorders that can have multiple genes impacting on multiple genes that include heart disease, diabetes, alcoholism, mental illness, and cancer. They can also have environmental impacts. So with the different ones that we just talked about, how much the genes are playing a role in these is still to be determined, because there's so much interplay between the genetics and your environmental, the environment that you are living in. So with the more truly past genetic disorders, genetic counselors are able to kind of help determine, such as with Huntington's, they can give information so that offspring can make informed decisions regarding their potential children. And again, they can use probabilities, they can use Punnett squares to be able to describe this. And as more information comes about, they may be able to make a more educated prediction as to what the potential offspring could have. So amniocentesis is one way we can find out information by taking a little bit of the fluid that surrounds the fetus. With chorionic villus sampling, CVS, you take a little bit of the placenta. We can also do physical observations through ultrasound. And there are many genetic disorders that can be ascertained now at birth by just doing a quick DNA sampling and doing a gene screen. So with amniocentesis, We are able to karyotype the chromosomes to look for any chromosomal abnormalities. You can also do that with the CVS, and you can run genetic tests as well. So this is just kind of summarizing those different relationships. We talked a little bit about this briefly. Alleles affecting a single gene, and then within 2 or more genes.